So, recording has begun. Let's get this over with. Let's talk about stuff. All right, so you read a bunch of Basidiomycete text material. This is essentially the last big textbook reading assignment that you have for the whole semester. There may be a chapter or two here and there, but mostly it's going to be papers from here on out. So, thoughts, questions?
I have a question about the quiz.
Okay.
It's one of the questions, question 8. I didn't know if you wanted us to jump into the quiz yet, but...
Yeah, why not? Sure. Let me pull it up. Okay. Question 8. Oh, yeah. Yeah.
So I
That's
was
right.
able to find the right answer from the textbook, but I had a hard time with the reading, understanding like why this was happening or why it was a quarter of the spores.
Okay.
So
you got the right answer, 250.
Yes, I got the right answer, just don't know the reasoning behind it.
Okay, so remember we talked about in the yeast how they have this like bipolar mating system where you can be A or alpha?
Yeah.
Right? They have essentially like two sexes, kind of in essence. In the majority of fungi, especially in Basidiomycetes, it's like 4. Okay, so you'll have 2 loci on your chromosome, and they tend to be kind of right next to each other on the fungal chromosomes, and they're hanging out there. One of them is mating type gene 1, the other is mating type gene 2. In order to successfully mate with another of your species, that organism, that individual, has to have different alleles at both of those loci, not just one. So it's not like saying male and female. It's 2 genetic loci that are controlling this. So in order to successfully mate, you have to be different at both of those. Does that help explain it?
Yeah, I'm just trying to wrap my head around that. It's kind of weird.
Yeah. So imagine a basidium, karyogamy happens, and there's independent assortment, meiosis, all that good stuff. We get 4 nuclei. On average, each of those 4 nuclei is going to be a different combination of both of those sex identity alleles. So they shouldn't be able to mate with each other.
Okay, I think that makes sense.
Okay.
Yeah.
And so, yeah, any 2 given spores, there's a 25% chance that they'll be able to successfully mate with each other.
Gotcha. That helps. Thank you.
Mm-hmm.
Hey, Dr. Zahn.
Yeah.
So it looks like I got question 11 wrong, but I was pretty certain I had it when we were talking about it, because I remember you were saying like that's the question about like the death mushroom. I thought it was called like the death angel or something like that. I remember you were saying that it was deadly because like it kind of had like 2 caps, like one halfway up the stalk or the stem.
It's got the ring around it. It's called an annulus, it's like the remnants of the veil. Yeah. So the question was for the genus name, not the common name.
So what was the answer to that one?
Yeah, so I still put a genus, but I must have put the wrong one somehow.
Amanita.
That's what I have, and it gave me 0 points for it.
Yeah, that's what I have.
But it's 0 out of 0.
It's 0. If you put Amanita, I'm going to give you a bonus point.
Okay.
Oh, okay. I was tripping because I was like,
it was worth 0. Canvas didn't give me a way to like include a bonus point that I could easily find, so I just made it worth 0.
Okay, cool.
Yeah.
Can we also go over 7? Oh, sorry.
Oh, go ahead.
I was just gonna
Okay,
ask the same question.
Oh, I was gonna ask about question 7, if we can go over it.
Ooh, yeah. This was another. I
yeah,
had
that
two
was-
really tricky
I
ones.
thought I understood that, and then I was very confused why it was the left, not the right.
Okay. So looking at this, you'd have to like either have a photographic memory or go back to the slides.
So, and I did. I went back to the slide and I looked in the book.
Okay.
And maybe
I
I just
took the-
misunderstood. I don't know. Maybe I'm just not taking it right about it.
I took the image from the slide and just flipped it to the left.
Oh,
Yeah.
okay. That answers everything.
The way you tell is that the little clamp connection forms towards the Spitzenkerper and then lays down that cell wall, and so that's what we're seeing here.
Okay.
This little septum right there.
That alone just explain everything. Thank you so much. So I was thinking the right way. I just looked at the image the wrong way. Got it.
Okay.
Okay.
Yeah.
Thank you
So
so much.
just to clarify, so whatever side the clamp's on, that's what side the Spitzenkerper is going to go on.
It's growing that direction. Yeah. And so this hypha is growing to the left. Yeah.
Okay. Can you also go over question 6 as well?
Oh yeah, internal spore production. So gastromycetes. Remember, they're not a real group, or they're not a monophyletic group. Okay, so is it derived or ancestral? If you look at the tree of basidiomycetes, they started out with external sexual spore production, right? Because they actually derived from ascomycetes, which do it externally. Most basidiomycetes still retain that, and so in a few cases they've independently evolved having their hymenium on the inside of their body, and so it's a derived trait. Yeah.
Okay, and then on, I think it was question 8 or 9, I asked about a sister group, and so it talked about basidiomycetes and ascomycetes are related, but then it talked about a specific group being like the sister group. But were you looking for the one that said this is the sister group, or were you looking for ascomycetes because it says they're related?
I'm looking for ascomycetes. Now, we didn't discuss any of the weird outliers, cystidiomycetes and these weird things that are currently placed as sister to Basidiomycota. If you put those, that's fine. I'll accept that also, but
okay, because in the textbook, okay,
yeah,
so, yeah, I was just confused because it said specifically like this is a sister group, so that's what I put because I wasn't sure if it, because it didn't clarify saying it was a sister group, Basidiomycete and Ascomycete. It just said they were related, so I wasn't really sure.
Yeah, and what this comes down to is like we've got Basidios, we've got Ascos, and then out here, Zygos. Let me bring this up.
Yeah, because I had the same question where in the textbook it talked about Basidio, Mycota, and Ascomycetes being the group that is sister to Mycota.
Yeah, and remember that's actually wrong in your textbook. If you remember from our lesson slides, we've now placed Glomeromycota deep within the zygomycetes. But here is, I don't know if you can see that. We've got our zygos. Basidios are sister to ascomycetes unless you start counting some of these weird little groups there, which we didn't cover in class, but if you put them as explicitly sister to the Basidios, that's okay. There's no necessarily one right answer to this question, because trees do tend to shift here and there.
So I put the one you said the textbook was wrong, but I didn't I forgot that we went over that in the slide. So if we put that, is that the wrong answer?
The glomeromycetes, is that what you put? Yeah, since we covered that one in lecture, that one is definitely not sister. There are some that might be sister to Basidiomycetes that we just didn't cover in lecture because there's, millions of fungi and we can't cover them all. But if you put one of those, that's okay. But Glomeromycota is not correct. It's not even a phylum.
Could we ever go over question 2 as well? Like I saw from the book and I kind of understood it, but I'm not 100% that I have it like 100% like understood.
Yeah, isn't this crazy? So this is how most Basidiomycetes eject their spores. So this is the spore. I just took this image from the text. It's a spore being ejected off of the sterigma. So you've got your basidium, the sterigma sticking off, the spore grows there, and then it's got to get off of there. Some of them don't eject their spores, but most do. What's happening here is the spore itself in the fungus starts pumping sugars into certain regions of the cell, high concentration.
Okay.
And those sugars, I don't know if you leave like a sugar bowl out, it doesn't quite work here in Utah, but in a humid climate, it gets wet, right? Salt and sugar is hygroscopic, it attracts water. And so it starts forming this little surface coating of liquid, and it really starts pumping tons of sugars tip, like the asymmetrical tip, and it gets this big ball of water. Okay, it's just, well, it's tiny, but it's a relatively large droplet of water.
Okay.
Now, when two droplets of water come together, what happens? It's like instant, right? They like move, they join forces really fast. Okay, that's hydrogen bonding at work. And so what happens is when this, it's called a Bowler droplet from your textbook, when it gets big enough to touch this film of water, they join forces nearly instantaneously, and that shifts the center of balance of this spore and launches it incredibly high acceleration just from shifting the center of balance. And so this process here is, if you put Bowler droplet or Bowler ejection or spore being liberated from the sterigma or active dispersal of basidiospore, anything like that, that's what I'm looking for.
Okay, perfect. That makes a lot more sense. Thanks.
Yeah.
So in the section C of that diagram where it has the little palms with A, is that the direction that the spore is being launched?
So these are actually physics terms, and we've gotten like physicists evolved into this, like Newtonian physics. You can see like this is the force here. This is the what's the word? Come on, it's been a while since I've had physics. It's not asymptotic force. Some A-word force. Basically showing the difference between the previous center of gravity here in the open circle and to where it moves when all the water is wrapped around. So this is basically like a force calculation. It's not really necessary for this answer, but.
Does it leave from like the tip or where the ball is at the bottom?
It shoots off this direction.
Okay.
Yeah.
And is this like the general mechanism that is used, or is there like many different kinds of mechanisms?
This is the main one, which is why it gets so much coverage. The other mechanisms for active dispersal in Basidiomycetes are pretty rare. If they're not doing this, then they're just not actively dispersing. Like a puffball, it doesn't do this because it's a gastromycete. It makes its spores inside, and it just relies on raindrops or children to come along and smash it and eject them with air pressure. Yeah.
Okay,
but this
thanks.
is very common. 99.999% of all the mushrooms you ever see are going to be doing this process right here. And so if you make a spore print by setting down a mushroom cap on some tinfoil, putting a cup over it, it's actually kind of useful to put like a little drop of water on top of this mushroom cap just to make sure that humidity stays up there so that there's a way for this Bowler droplet to grow. And so the spore print that's left over is the result of millions of these little bowler droplet mechanisms happening. Any other questions about the quiz?
Yeah, question 1.
Okay. Basidium. Did you put something different?
I'm sorry, it cut up. What did you say?
Oh, Basidium.
Oh, Basidium. Okay. Yeah, I put gill.
Oh, yeah. Yeah.
Okay. That makes sense.
Okay. Okay.
So I have a question about the reading.
All right.
If we're done with the quiz. So they talk about the dolipore septa. I don't know if I'm saying that right. The parentosome or something like that.
Mm-hmm.
Is that basically the equivalent of the Warrenian body that we're talking about with the ascomycetes?
Yes.
It is.
Yeah.
Okay.
Mm-hmm.
It's just a bit of different structure, but same.
Yeah,
same
and
thing.
it uses different proteins and everything,
Yes.
so cell biologists gave it a different name. It does behave a bit differently, but functionally serving the same purpose. Yeah.
Okay. So this is basically the whatever the basidiomycete version of that.
Of the plug. Yeah.
Of the plug. Yes. Awesome. Thank you.
Mm-hmm.
So I've got a question.
Dr. Zahn. Oh, sorry.
Wait, Austin was going first, I believe.
Oh yeah. I'm not sure if it's question. Did we go over either 8 or 9? I think the one talking about gastromycetes and like what it refers to.
Oh, in the quiz?
Yeah. I'm not exactly sure which question or how it's worded, but
There
I was
were
kind of
a
confused
couple
on
about
that, like
gastromycetes.
like the phylogeny of gastromycetes and what it refers.
Oh, it was
It's question 3.
the paraphyly in the gastromycetes.
Should be question 3.
Maybe question 3.
Yeah.
Yeah.
Because basically I'm asking, what does the term gastromycetes mean? Right? Why do we make this into a group even though it's paraphyletic? And it just gastromycete refers to internal spore production.
Oh, okay.
Yeah.
Okay, so you weren't looking for like a whole explanation, basically just what is another name for gastromycetes?
Yeah, kind of. What makes them a group, even though they're not monophyletic? Like, why is it that people decided they were a group? It's because they all produce spores internally. Yeah. Mycology, unfortunately, is historically full of really bad accidental choices in how people made groups based on morphology, because so many traits have been like independently evolved that look the same. So many traits are lost, and so we pick things like, oh, if they've got zoospores, they're a Chytrio mycota. If they make spores on the inside of their fruiting body, they're a gastromycete. And these were just kind of unfortunate choices back in the day by people who started as plant biologists, where it kind of worked out well, and so they just kept with it.
Yeah. Okay.
Mm-hmm.
Thank you.
So for that same question, the answer that I got from the reading was that the thing that differentiated them was that they don't discharge their basidiospores violently.
Yeah, that'll work.
Okay, that works.
That'll work. Technically, they got the name gastromycetes because of like gastrointestinal, right? So it's like the inside of the body, and so that's really what it refers to. There are a few random basidiomycetes out there that don't discharge their spores, but also aren't gastromycetes. But that's okay. They're few and far enough between that I'll take don't discharge their spores violently is an answer for that.
So I have a question from the reading.
Okay. Yeah.
So in chapter 18, it talks about the asymmetric expansion of the basidiospores. Is there a specific reason why they don't grow symmetrically, I guess?
Yeah, and it goes back to that bowler droplet. If they were
Okay.
symmetrical, they wouldn't have that ability to shift their center of balance super fast. And
Okay.
so it is adaptive for them. It helps them with their chosen method of violently dispersing. Being asymmetrical gets you a much better dispersal than being symmetrical.
Okay, that makes sense.
What would you... Oh, Hayden, are you waiting for a question? Anyway, what would you want us to know about clamp connections?
What their purpose is and where they're found.
Okay.
Where, as in phylogenetically, where they're found, right? So if you see one, you know you're not looking at a zygomycete or an ascomycete.
Yep.
You know you've got a basidiomycete.
They're
But
only in
even more than that, you know something extra about that basidiomycete. What about it? When do clamp connections happen in their life cycle?
Oh, when they're making basidiospores?
Oh.
Oh, plasmogamy? Right?
Yeah, so after they are dikaryotic.
Yeah.
So if you see clamp connections, you know that it's a basidiomycete. Not all basidiomycetes make them, but only basidiomycetes do. And you know that it's a dikaryotic mycelium because they can't make them if they're haploid. Yeah.
Okay.
So if you're a geneticist, trying to figure out something and you are hoping that you've got haploid mycelium, there's really no need to do like chromosome sequencing and counting and transmission electron microscopy. You can just look for a clamp connection. If you see it, you know you don't have a haploid mycelium. So it's a nice little clue. Yeah.
Back at the beginning of the semester, we watched a video about like how fungi changed the world. It was like the brown rot and like the white rot he talked about, like the same as Basidium mycotus.
Mm-hmm.
Okay.
Yeah, those are only basidiomycetes, the brown and white rot. We'll see them next week because we're gonna briefly look at the ecology, like functional ecology and physiology of fungi, and we'll see like what brown and white rot actually means. Whether it's eating lignin or just cellulose, that kind of thing.
Okay.
So I had a question about puffballs.
Mm-hmm.
When their spores are released, they're like they have to be like broken open, right? So after they release their spores, do they repair their wall, or do they die after that then?
It's done. The fungus doesn't necessarily die because remember, like the actual body of the fungus is underground or in the log or whatever. That's 90% of the fungus. We just never see unless we dig underneath. The mushroom or the puffball itself is akin to like an apple on an apple tree. Right? It's just the fruiting sexual structure. And so you can pluck that off and it probably isn't going to harm the underlying fungus very much, just like picking an apple doesn't hurt the tree. So yeah, once it's done its job, it's over. It will like close off that section, right? And quarantine it, and it's done. It'll try to go dormant for the winter or whatever, and move on to fight another fight.
Oh, okay. Thank you.
Along those same lines with the puffball, you said that raindrops actually can activate them to mist out, right?
Yeah, totally.
So
it doesn't take much.
Okay, and then you also said that I'm trying to remember from last class, did you say that all of these spores have to go through like a winter cycle before they can actually grow again, or?
No, for the rust fungi, the
Oh,
ones
rust
that,
fungi.
that annoying life cycle,
Right. Okay.
those do tend to require some period of dormancy. Whether in most cases it tends to be winter, but there are rust fungi that like live in the tropics also. Obviously, there's no winter there, but they do need like a dormant period.
Okay. But the puffballs, they don't need a dormant reference
No,
period.
no, those spores are good to go.
Okay.
They're ejected. Yeah.
Okay. Thanks.
Dr. Zhang, can we go over the like when vegetative incompatibility happens and kind of what it means as well? I mean, I
Sure.
think I know from the reading, but it would be helpful to hear your explanation. And then
Okay.
maybe can we actually go over the rust life cycle one more time?
Yeah.
Make sure we have
No
the
problem.
Thank you.
It takes a while. I understand. So vegetative incompatibility is essentially, it's pretty useful to keep a fungus genetically pure, so to speak, because if you're dikaryotic, you want to have two different versions of nuclei in you. You don't want identical nuclei and to call yourself dikaryotic. That's not very useful. Some fungi can get away with that, and the two nuclei wind up in the same cell and they reach that critical threshold of making their secondary compounds or whatever, and it can go and make a mushroom. But by and large, vegetative incompatibility means that one hypha is growing along, another one next to it in the same fungus. That hypha can't join up and do plasmogamy with itself. Does that make sense?
Yes, it does a lot. Okay, that
Okay.
helps. I'm more clear than
And so
from your
essentially, it's a mechanism to make sure that you're not doing plasmogamy with yourself.
with yourself. Got it.
Yeah. And they accomplish that by a lot of like weird little genetic tricks, surface proteins, that kind of thing, that can vary. And sometimes it can go wrong. Like you can trick a fungus into melding with itself. You can get their hyphae to connect with each other, but you have to like alter their surface proteins or do knockout genes on some of those. But in nature, it's not very common.
It seemed from the book, however, that you were saying that if it comes in contact with a hyphae that has the same nuclear genetic information, then it is fine to do plasmogamy and it can continue and be just fine. Okay.
Yeah.
Okay. So making sure I understood.
And when they accidentally do plasmogamy with themselves, it usually just dies. Like that little section, they seal the bulkheads and that little section turns brown and dies off.
Okay.
So it just kills off part of its body. It's like, oh, wait, I accidentally am trying to mate with myself. No good. Yeah.
Thank you very much.
Mm-hmm.
Kind of a little bit more clarification about the vegetative incompatibility. So it doesn't fuse with itself. Is it also so it doesn't fuse with a different species as well?
Yeah.
Okay.
Yeah. It needs certain signals in order to say, hey, this is my same species, but also not myself.
All right.
Yeah. Let's see here. And I promised I'd go back over the rust life cycle. So before I forget. Okay. Down at the end. Okay. So the key to the rust life cycle, there's a few, but the main one is that they really, really depend on living plants. They can't live without their host plants, and they've got two host plants. So part of their life cycle has to take place in one plant and the other part in the other plant. This isn't like super weird if you know anything about malaria, right? it's got to spend part of its life cycle in a human, part of its life cycle in a mosquito. So it's the same kind of thing, but these are two different plants. And there are two main stages. So the ecial stage is where they do this sexual recombination, okay? And they become dikaryotic. So this is akin to, like we were just talking about, a fungus like meeting a compatible mating type hypha, and then they join up and plasmogamy happens. For the rust, they do the same thing, but instead of two hypha meeting up, it's a little spermatium, which is just like a little tiny chunk of hypha, meets up with a resective hypha on another individual. Okay, so it's just like they're meeting up in the soil or whatever, functionally, but the mechanism is quite different. Here, they got to make it through some air, essentially, and so they're using insects generally for that.
And this is we're in the leaf, right? We're on
Yeah.
top of. Okay.
Yeah, so this is showing the leaf here. It makes a tiny little body, and all this is good for is making these little gametes, essentially, and receiving gametes from another one. So it's almost like a little flower in that sense.
Yeah.
And you can think of the spermatium kind of like pollen. Okay, so once the spermatium from a compatible same species makes it to another little body, another individual here, if it lands on this receptive hypha, now we can do karyogamy or plasmogamy, sorry, and it donates its nucleus. And so as you can see here, I can blow it up a bit. Well, they've got two different nuclei in each one of these spores. So it's dikaryotic at this point.
Right.
Okay. So it goes dikaryotic, and like any other basidiomycete, once it's dikaryotic, it induces a bunch of changes. And it's like, okay, you're going to live a long, healthy life now. You can make massive mycelium. You can make fruiting bodies. So what it does is it starts growing hyphae through the leaf to the underside of the leaf. Okay, so then it makes something called an ecium. And the entire function of this little thing is to produce eciospores. Okay. So these eciospores go and they land on the second host. All right. So in this case, a willow. They land there and they start killing off the willow slowly, but they're just using up its nutrients and everything, growing hyphae through its cells, and make uridinospores. So these will be like little, it looks like little blotches on the leaves. In fact, a lot of these are rust colored, therefore we call them the rust fungi. Okay. It can reinfect the willow over and over with uridinospores, but when either the tree's about dead or those leaves are about dead or winter's coming on, it will produce this like resting structure. And we call that a telium, or the teleospores. Okay, those are the things that overwinter. They get thrown off of the willow leaf, they land in the soil to be kicked back up in the spring. Okay. The telium itself is a basidium in essence. It's just like a resting basidium waiting for the chance. It makes basidiospores that then go and infect the first host again, and the cycle continues.
So basically,
I don't know if that helped.
Yeah, no, they helped a lot. But the iridium and the thallium, basically, they're the same thing. We just call them differently at different stages, correct?
Yeah, the uridinospore is genetically the same as the teleospore, but the teleospore is the resting form. It's got like that thick cell wall around it. It's ready to just hunker down and wait for good weather.
For good weather.
Yeah.
Okay, got it.
Mm-hmm.
Thank you. I think that's where the confusion was.
Okay.
At least for me, that helps. Thank you.
Yeah, if you just added like a really thick wall around a uridinospore, you basically got yourself a teleospore.
Teleospore.
Yeah.
Thank you so much.
No problem.
All right. Unless Benjamin has a question, I have a clarification question.
Okay.
Okay.
You're good.
So clarify something for me. We just talked about resting stage spores that can overwinter.
Mm-hmm.
But at some point, we've also talked about how in Basidiomyces, if they don't get another nucleus, they die off, right?
Yeah.
Which is the opposite of being able to last for a long time. So at what stage are they vulnerable to that? Is that when they start initially growing as hyphae?
Yes.
Okay.
Yeah. So the spore, and keep in mind this, maybe there's a bit of confusion here. That Basidio spore from the teliospore. So this thick-looking structure here is the teliospore. It still has, it's dikaryotic.
Okay.
Like through the winter. So it's dikaryotic. It undergoes meiosis or karyogamy and then immediately meiosis in the spring. So it stays dikaryotic in its resting form. Spring comes, there's larches out now, and so it wakes up, undergoes karyogamy, and then immediately meiosis, and it makes those basidiospores, those haploid ones. Now, they're good enough to make one of these structures, these spermagonia, but that's about all they can do before they die off. If they don't get another nucleus from an adjacent spermagonium, like that's the end for them. In other basidiomycetes outside of the rusts, it's once they start making that initial hypha, we call it like a primary mycelium sometimes. So the basidiospore lands on, I don't know, a cow pie, and it's like, woohoo! It wakes up, it starts sending out a mycelium. It's not very big, it doesn't branch very much. It's essentially just searching for a compatible mycelium at that point.
So do ones that aren't rusts, your typical ones, do they have like teliospores where they overwinter, or how do they do they have types of spores that allow them to last a long time without becoming primary mycelium?
Yeah, they just don't get the name teliospore because their spores are haploid.
But if they're haploid,
yeah, well, it's a basidiospore. The teliospore is dikaryotic. It's not haploid. And
okay.
so when it does its resting thing, since it's a rust and since it's dikaryotic, it gets its own stupid name. We call it a teliospore. There are basidiospores that are haploid that can last quite a while. They can overwinter. One of my recent research projects was at the Mauna Loa Observatory in Hawaii, and we were pulling in like upper atmosphere air samples over 13 years, and we found these things like floating around intercontinental up like with the baking UV rays and everything, and they were still viable. So they're okay.
All right.
It's just they're haploid. And it's a species by species thing, right? Like how thick the cell wall is. Do they have a melanized cell wall that helps prevent UV from damaging their DNA? So there's a lot of characteristics that go into it. Some species don't have like a nice resting spore stage. Those tend to be in warmer climates, tropical regions, that kind of thing.
Okay, thanks.
Mm-hmm.
How big of a difference does the melanized like outer casing on them help with like the UV rays? Is it like a huge difference?
Huge difference. Yeah.
So the ones that you found had melanized outer shells on them?
Yeah, well, we found clear looking, they're hyaline spores also, but couldn't get them to grow. So they were probably just fried by the sun. Yeah. They're still floating around out there, but doing nothing except, causing ice nucleation or forming clouds or whatever. They're not viable fungi anymore. Yeah. Melanin is a very useful biomolecule. And they use the exact same melanin that humans do in our skin. Which shouldn't be too surprising given that we're both episcons and not that super different from each other biochemically.
Hey, Dr. Zahn.
Yeah.
So I'm sorry I showed up a tad late. I just got out of an interview. But is it too late to ask a question about one of the quiz questions?
No, we can go over it again.
While I was taking the quiz, I had a question about vegetative incompatibility.
Uh-huh.
Reading in the book and then also like looking up different sites, something said like it didn't seem conflicting, but it was like, okay, what would you say is vegetative incompatibility?
The inability to perform plasmogamy with your own self and have it survive and donate a nucleus. So
with your own self, wouldn't that be it's compatible because it's with your own self? Maybe that's where I was going wrong.
Oh, so the incompatibility part is like if a hypha is growing along and splits,
yeah,
and then grows back in on itself, is it able to then essentially donate a nucleus successfully and become dikaryotic? Incompatibility says no. That if it does manage to do plasmogamy, that little section will get shut off and die.
It'll do like programmed cell death if that were to happen.
Yeah, pretty much.
So then what's the benefit of having incompatibility versus compatibility?
Well, if you were able to mate with yourself,
that'd be a little weird.
Well, weirdness isn't really the issue.
Okay, fair enough.
All right. Selection cares about. It would be weird, but what would your offspring have?
You would give half of your DNA, and then you would also give the other half of the DNA.
It'd just be reshuffled, of course, but so they wouldn't be a clone of you, but they would have all your same propensity for genetic diseases and everything. It's like inbreeding times a million if you were able to breed with yourself, right? And so if this doesn't exist in nature very often because it does tend to build up genetic diseases, that kind of thing.
Gotcha. Okay. No, that makes a lot more sense.
Okay.
Thank you.
So aside from questions specific to the quiz or the reading, we're about to do our first exam. So now's a good time for general fungal questions. What's still kind of murky to you about fungi or the groups that we've looked at? What's not clicking?
To be honest, just separating out all the specific terms that are for each group that we've gone over. I feel like every time we do a new week, and it's a lot of reading, and it's a lot of new terms.
And they're all different from the previous terms.
And then it's like I go back and I look at zygomyces, and I'm like, oh my gosh, I know I read all of this, right?
Yeah.
But I did not retain as much of it as I thought I would because my brain's just like, well, you just learned a new 80 terms, right?
Yeah. What to say to that? How to address your question without being totally depressing? That's what I'm thinking right now.
Get good and get some flashcards, it sounds like.
Well, no. Don't place too much emphasis on all the terminology. It comes with time and with repetition. And so I wouldn't spend, 3 hours a day with flashcards memorizing these terms. Look at the big ones that I bring up in the lecture slides. So if I say sporangiospore, you should probably have a little flash of, oh, that's like a zygote thing because they make sporangia. We saw that on our bread mold, right? If I say ascospore, that one should be pretty straightforward to you. That one's in ascomycetes, okay? So you start getting used to kind of the root of the word and the little stuff they tack on. Basidiospore, ascospore, zygospore. Those are simple enough. Then, of course, you get into the rust fungi, and it's like, oh, what's the difference between an esiospore and a teleospore? And all I would say is that you do get to look that up during your test, and you'll have a week to take it. So
For the test, is it going to be like the retrieval practice where we can come back to it, or is it like do it all at once kind of a thing?
Oh, I'm giving you a Word document. Yeah, so there's going to be a Word document with blank spaces, and you fill it in blue text so I can like easily see what you've written, and then upload that to Canvas.
Oh, okay, cool.
Yeah.
So for these tests, are you breaking them up into the different sections that we've gone over, or are they kind of just all meshed together?
It's all meshed together.
Okay, good to know.
Yeah.
So these Word documents, are you talking about the kind of like the midterm number 1 that's on the, it says it's on the 25th?
Yeah.
Oh, okay, cool. Just
So
wanted to make sure it's on the same page.
Yeah, so you'll upload it to Canvas. I think it's just exam 1. It's due the 25th. Just upload your docx file after you've completed it. Put your name on it or whatever. Not that it matters because it'll be associated with your account. And then you're good to go.
Okay, cool.
Yeah, and I've got instructions on there, but people don't always follow those. So let me go ahead and tell you. It is just a Word document. It's got numbered questions. You know, I'll put like some pictures on there and say match these to the phylum where they belong. Okay. So for picture A, what phylum does it belong to? You just write that in there in nice blue text. Okay, so it's easy for me to read because I'm gonna have a bunch of these. I may ask you something about like how to define a fungal species in the 21st century, right? And so that's 3 or 4 sentences you got to type in. Just type it in at that question in blue text. Okay, save your document when it's done, upload it to Canvas, and you're good to go.
Awesome, thank you.
And just another reminder, I know we covered this a little bit, but you can work with each other on this if you want. Okay.
How long do you think it'll take us?
I'm not
That's scary.
finished with it. I've got, it's 100 points. I've got 78 points worth of questions right now. I think at this point, if you know what you're doing and don't have to look stuff up, 20 minutes. If you have to look stuff up, that kind of depends on you.
Okay.
Yeah. It's not meant to be super hard. I'm treating, in my upper division courses, I don't treat tests as like a kind of a got you situation to see if you can recall a bunch of material. I try to treat exams as just another learning opportunity. And so that's why you can work together, you can use the internet, that kind of thing. It's just like a refresher for you to go through and kind of like practice recalling this information. And some of these questions you're going to be like, oh yeah, I know this, and you're going to feel really great. Some of them you're going to go crap and have to look it up and still not quite get a direct answer. And so it'll be a learning process for you to answer that question. And so in essence, it's to help you find out what you're still kind of lacking in terms of mycological knowledge that I expect at this point, and not to punish you for not knowing it yet, but to give you a chance to look it up.
Okay, sweet.
Hey, Dr. Zong.
Yeah.
If we're doing pretty good on these quizzes, we should be doing pretty good for the test, right?
Yeah.
Okay.
Mm-hmm.
About how many questions are on the test, do you know? Or I know you said you have about 78 points, but
There'll be about 30, 35 questions.
oh, that's not too bad. That's good.
Yep.
And then did you say when it's gonna be post? I know you said we are gonna have a week, but I don't know when we're gonna have it available.
I will finish it today. I'm pretty close, so I'll just finish it this afternoon.
Okay.
And immediately post it up on Canvas, and I'll put a notice here on Teams letting you know it's available. And so you'll have from when I get it published until the due date.
Okay. Sweet.
So unrelated to the exam, but for collecting mushrooms, where would be a good place to go, like hiking, to find stuff?
Any of the canyons. You don't want to be out on like exposed slopes covered in sage. You know, you want to be in shady areas near water. Trees are important, especially for basidiomycetes, because a lot of the trees here in Utah actually depend on ectomycorrhizal basidiomycetes, and they make nice, pretty mushrooms. So looking around trees near water sources, find fallen logs. Most mushrooms are saprotrophs, meaning that they like need dead plant material, and so leaf piles, rotten logs, that kind of thing are going to be your best bets.
I do have a tip for that. For my entomology class, we went down to, I think it was like Hollow Springs Canyon or something down in Springville, and there was a bunch of, I think they were oyster mushrooms, the big flat ones.
Yeah.
Growing down there in the park. So if you need those.
Sweet, thank you.
And be careful if you're on like state land. If there's a ranger around, they're gonna get weird if they see you picking stuff from state land. So just keep that in mind. And they don't understand that picking a mushroom doesn't actually harm the fungus, because they think that is the fungus. They don't know that it's growing underground. So just be aware of that.
What about like yard mushrooms? Is that okay? That's like, and we wanted 5, right?
Yeah, 5.
And no yard mushrooms, you said?
No, yard mushrooms are okay.
We are fine. Okay, good.
Yeah, I don't care where you find them. Just basidiomycetes. Honestly, if you want to go to the Asian market, they've got like 6 varieties of basidiomycetes hanging out in their produce section. If that's what you want to do, it really is. I can't expect. Well, can't expect everyone to like, have a car and have time to like go drive somewhere and do some collecting or whatever. So that's an option. And the thing is that you're going to be filling out that sheet for them all. So it's to get you to look at them closely and like learn to describe them. Of course, your photo of the mushroom in situ, if you go to the Asian market, is going to be very different than if you actually found it under a tree. I'm going to make you take a picture of where you found the mushroom. And if that's the produce aisle, I want a photo of that. That's reality.
In its natural habitat.
In natural habitat. And the benefit there is you can like saute it in butter afterwards and have a nice meal.
If we find any mushrooms that we think are the ones that you mentioned are edible, can we send you a picture for verification purposes?
I will need a picture of like a slice down the middle so I can see how the gills are attached. And I will need a picture of the spores and the spore print.
All right.
Yeah.
So is this the last section for this test that we're covering? Or do we have another section that we're covering?
Oh, no. Just weeks 1 through 4.
Okay.
Yeah. So this test is only over the groups that we've covered, right? So chytrids, zygos, ascos, and basidios. And so it's focused on, what those groups are, how they're related, can you identify characteristics of them, their life cycles, that kind of thing.
Okay, thanks.
Yeah, after this week, we're moving on. You can see, right? So we're going to look at, some mushroom ID stuff, right? So that's the lab assignment we were just talking about. So macrofungi, we're going to look at active dispersal mechanisms. So we'll get into what's actually happening in that bowler droplet, how mushrooms grow and how they grow so fast, because it's really freaky. People used to think it was witchcraft. You'd get the little fairy circle in your yard after a rain. They called it that because they thought fairies had come and danced there over the night. Because they go from 0 to 6 inches tall in just a couple hours, and that's crazy fast growth. So we're going to kind of see how that works. And then medical mycology, then we'll look at plant pathogens, so basically like human pathogens, plant pathogens, and then population ecology, and then we'll have another exam on those 4. Okay. Then we get into symbioses, and then look at ecosystem services of fungi, how they fit into like biogeochemistry. Then ethnomycology, where we talk about all the fun mushrooms that people illegally eat, and how people have used fungi over the years. And then it's bioinformatics of fungi, some like pretty brief stuff for bioinformatics, and then that's it. That's our course.
So for the identifying and finding fungi assignment, dried fungi, are those like not viable for that, or does that ruin their structure too much? But
typically, yes, it can be very difficult, especially if they're like kind of old. A lot of times fungi, they release their spores over like a 6 to 8 hour period, and then they just kind of start senescing. They start getting old. Slugs come along and start eating them. Other fungi, mycoparasitic fungi, will come and start eating them. Slime molds come and destroy them, and they turn goopy and disgusting and stinky. It's really hard to see characteristics when a mushroom is old or dried out. And since we don't have access to DNA sequencing this semester, we won't be able to identify those.
I was thinking, if you bought it at the store and it was dried, you would know what it is.
You would know what it is, yeah. But it'd be hard for you to identify stuff about the cap, right? Is it slimy or dry?
Oh, right.
Yeah. Are the gills attached or decurrent? Oh, well, that kind of goes away when it's all shriveled up and dried and cracky. Yeah, so even the morphology is really tough to describe from a dried mushroom.
Do you know if the snow mushroom grows in Utah? I was reading about how like it's in a lot of skincare products and it's like good to rub on your face. Is that
What is the snow mushroom?
It's part of like the tremella fungi. I don't know. I was just looking.
Oh, yeah. These grow all over the place. Yeah, this is one of those dacrymycete fungi. So they've got like the tuning fork basidio with just 2 basidiospores. And yeah, look for wet, fallen logs. So near streams, in the shade, you'll find these.
Okay, cool.
It can be kind of tough because it's pretty dry here in Utah even right now. Like we don't have a lot of moisture in the air for another few weeks probably, and so they look like these dried, shriveled up things, and only after a rain or a snow do they like start blossoming and look really pretty and jelly. Yeah, but yeah, these are used. Traditional uses, and we'll see this in like our ethnomycology section, but you can just smash them up and like rub them on your skin, and they work like aloe. They're super high in vitamin D. You can eat them. You put them in soup. They're actually quite good. They're kind of like crunchy, slimy texture. If you're into crunchy, slimy, they don't taste like anything. They taste like whatever you cook them with. But yeah, you'll see these in Utah for sure.
Dr. Zahn, when we look at fallen logs, do we look on the underside of them? Like, do we kind of like move them and look under it, or?
Well, with great care, given that snakes are a problem, always don't roll it towards yourself. Roll it away from yourself and look under. But yeah, if you go on a hike with a mycologist, you'll spend 3 hours and maybe go like 300 yards because mycologist hikes are just the nerdiest thing ever. It's people crawling around. Yeah.
That's not what I thought. I just wanted to like make sure I'm not, looking stupid for no reason.
No, you're looking stupid for a good reason.
Okay, great.
Yeah.
Awesome.
So, Dr. Zahn, would
Yeah.
you be up for doing like a field trip for finding fungus?
No.
Okay.
Short answer. But let me see. Utah. Oh, forget the name of the group. I went and gave a talk to these folks. Mushroom Society of Utah, there it is. So you can hook up with these people, the Mushroom Society of Utah, and they do guided trips almost every month. And especially right now, they're going to be like going out for it. So let's find events. That's out of date webpage. Their Facebook page. It's probably the place to go to see like up to date info. Fall foray, August 29th. So you missed the big fall foray, but to me, August 29th is still summer, so whatever. They might be worth contacting, and they have a lot of folks in that group that are really good at identifying mushrooms and know good places to look. And even if they're not doing a big foray, they may hook you up with somebody who is going out, this weekend or something and might be able to show you a good spot.
Okay. And there's been, some fires around, right? Would that be a good time to find, what are they, morels that people like?
Yes. Not, directly after a fire, or especially not where it's still potentially active. You need to get out of the way of our professional firefighters. A lot of times, morel hunters really do more damage than they ought to because they're in the way. They're parking in front of gates and stuff where firefighters need to be. But for a fire that's like 3 to 4 weeks old, yeah, go check it out. There's a good chance that you're going to find morels. But morels do not count for this assignment. How come?
They're not Basidio?
Yeah,
they're ASCO, right?
Yeah.
All right, so we need to be finding, so it's not any mushroom, it's Basidiomyces. And why do people like them? Are they just very tasty?
I think they're pretty overrated, honestly. They are good, but I don't think they're like tasty enough to warrant the insanity that they arouse in people. I've had a gun pulled on me because it was somebody's like private morel patch out in public land. It's not worth it to me. They're not that good. They're pretty good, but not that good.
Is it also because you can't buy them in stores because they're sporadic when they grow?
Yeah, it's a scarcity thing. And to eat them, you fry them in butter. If you fry anything in butter, it's going to taste good. Escargot is delicious. It's boiled in butter. Like, it's awesome. So they taste like delicious garlic butter. So they're definitely worth trying, but I wouldn't like plan your month around morel season or anything. One of my favorite mushrooms to eat is the oyster mushroom. I think it's absolutely delicious and has excellent texture. Very easy to grow at home, easy to find out in the woods, and cheap at the Asian market. That one in Orem, Asian Market Orem, it's like 800 North on State Street.
Which
Great store.
mushroom did you say?
Asia Market Orem.
No, what mushroom? Sorry.
Oh, the now I'm brain farting.
Oyster.
Oyster, thank you.
Oh,
Yeah.
oyster mushrooms. Okay, sweet.
Yeah, and they've got there 3 varieties of oyster mushrooms. They've got shiitakes, they've got
which are delicious.
yeah, flammulina, with straw mushroom, or enoki if you've been to a sushi restaurant. So lots of good mushrooms can be found there. If you're interested in trying out some unique edible mushrooms and don't feel safe finding them in the woods. With that said, when you do find an oyster mushroom and you get confirmation that it is a oyster mushroom, you'll never confuse anything else for it. And so it's my go-to for like wild edible mushrooms. All right. Any other questions?
Yeah, just one last question. When you got the gun pulled on you, did you proceed to pick them, or what happened?
No, I left. Yeah.
That's all. I mean, I figured, but I just had to ask.
I said, I'm very sorry, sir, for intruding on your private stash here.
Public land. Yeah.
Yeah, I didn't mention that my tax dollars went to pay for this land also. It was
Exactly.
It didn't seem like he was a reasonable sort of person.
Well, if you're pulling a gun on a mushroom, yeah, I could definitely agree that seems a little excessive.
Yeah.
I mean, the man loves his mushrooms. What can you say?
Well, it's big business for some people, right? Because they sell them to restaurants and grocery and stuff. And so, yeah, the people who are doing that aren't there because they like morel mushrooms. It's because they make a few $1,000s every year with it.
You had my curiosity, but now you have my interest. Oh, how does one get into this business? I got student loans that need paying off.
Utah's not a bad spot for it. If you go up to regions with decent snowmelt the year before that had a fire, about 3 to 4 or 5 weeks after that fire, you often get a flush of what are called black morels. And you can pull in, 50, 60 pounds of those in a day and make a lot of money. Yep. I'm sure there are restaurants in normal years, maybe not right now, with plenty of clientele in Salt Lake City that would pay top dollar for black morels.
And I can say, hey, I'm in a mycology class. I know for sure these are the real deal.
Yeah, don't quote me on that though. I don't want to go to prison.
According to
manslaughter or something.
I got a great reference here.
Yeah, right. No, I did have a mycology student who went and picked about 40 pounds of false morels. They look like morels if you don't know what you're dealing with, but they are false morels and they are poisonous. And so he sent me a photo and was like, you want some of these? I got about 40 pounds. It's like, dude, no, you need to get rid of those like immediately. He'd already given them out to like 8 or 10 people, saying that they were edible. So I had to get a list of all those people and basically contact them and say, don't listen to this person. You're going to die if you eat those. So do not quote me on safety of anything, except maybe oysters.
Mycology is turning out to be way more wild than I would have expected, for sure.
You should go to a mycology conference. Yeah, it's an interesting mix of like really truly crazy people, and old hippies that are super into mushrooms. It makes for a fun, like, scientific meeting, for sure.
Dr. Zahn, have you ever heard of the Seek app?
Of the what?
It's an app called Seek, S-E-E-K.
No.
So it's an app I have on my phone where basically I can take a picture of a plant or animal and it tells me the species, genus, all that stuff. I was just wondering if you'd heard of it.
I'm familiar with iNaturalist, which kind of is trying the same sort of deal.
It's by the same person.
Oh, okay. Yeah. That can be pretty good. It can at the very least show you where things have been found. It kind of, their goal is to really build a map of like species distributions. And a side goal is like to use a neural network algorithm to like try to predict genus and species just with a photo. I can promise you it doesn't work with fungi.
Oh, really?
Yeah. iNaturalist part is awesome. You can see like where it'll show you a picture and like where this thing was found, and people will give you their opinion of its genus and species. But the photos of mushrooms, that just doesn't work. We need to look at spore color, right? Like gill attachment to the stem. We need to cut open the stem and see if it's stringy inside or hollow. Like photo just won't get you there for macrofungi. Yeah, and mycologists, we get this a lot, like people sending us a blurry cell phone image from 4 feet away of some mushrooms with the question, "What are these?" Like, looks like a mushroom. Yeah, you got it.
Like some reference pictures.
Yeah.
Like they don't totally look the same as the one I took a picture of.
Yeah. So be careful with those apps for identifying mushrooms.
Okay.
They may get you close, but definitely not for deciding whether something's safe to eat. All right, with that, unless there's anything else, I'm going to go finish this exam and get it posted up on Canvas for you. All right.
Thanks, Dr. Zahn.
Thanks, Dr. Zahn. Have a good weekend.
See you next week.
Yeah.
Thanks, Dr. Zahn. Appreciate it.
Thank you.
Have a good one.